Our brains are constantly processing information, which requires communication between billions of nerve cells. Signals are passed from a sending neuron to a receiving neuron at a junction called a synapse. An action potential in the sending neuron travels down the axon until it reaches a synaptic terminal. The narrow gap between the synaptic terminal and the receiving neuron is called the synaptic cleft. The synaptic terminal of a sending neuron contains numerous vesicles filled with neurotransmitters, chemicals that carry information across the synaptic cleft. When an action potential reaches the synaptic terminal, the vesicles fuse with the plasma membrane of the sending neuron, releasing neurotransmitters into the synaptic cleft. The neurotransmitters affect the receiving neuron, changing the distribution of charge across its membrane. Let's take a closer look. An action potential is propagated down an axon by the opening and closing of sodium and potassium channels. When an action potential arrives at the synaptic terminal, it causes the opening of calcium channels, shown in green. Calcium ions enter the synaptic terminal through the calcium channels. Calcium ions bind to the vesicles containing neurotransmitters. This causes the vesicles to fuse with the plasma membrane of the sending neuron, releasing neurotransmitters into the synaptic cleft. The neurotransmitters diffuse across the synaptic cleft and bind to receptors in the plasma membrane of the receiving neuron. Here, the receptors are ion channels that open. Ions move across the membrane, changing the distribution of charge across the membrane. The neurotransmitters are quickly removed from the synaptic cleft, ending their effect on the receiving neuron. A single neuron can receive signals from many sending neurons. The signals arriving from the axon on the left are excitatory. They make the receiving neuron more likely to generate an action potential, as indicated by the green glows. Signals from the axon on the right are inhibitory. They make the receiving neuron less likely to generate an action potential, as indicated by the red glows. The two sets of signals cancel each other out, and no action potential is generated. Now, only excitatory signals are transmitted, and the receiving neuron generates an action potential down its axon. The signals that pass between these and countless other neurons constitute our thoughts and coordinate our activities.
Welcome to another Anatomy and Physiology Smart Art video, where we guide you through an important piece of art. After watching this video, you should be able to describe the 5 steps in a reflex arc. Notice we have 3 different kinds of lines to show what's going on. A red line is a stimulated sensory neuron. A white line shows an excitatory interneuron, and a black line shows a stimulated motor neuron. Let's start on the left with step 1, stimulation of a receptor. Here, the stimulus is that you touch a sharp tack, which stimulates pain receptors in your hand. Now, let's move clockwise to step 2, activation of a sensory neuron. Your stimulated pain receptors shoot action potentials to the posterior root ganglion and then to your spinal cord. Notice that this red line indicates a stimulated sensory neuron, and also notice here the cell body of the sensory neuron is located in the posterior root ganglion. Step 3. The information travels to your CNS for processing. Excitatory neurotransmitters are released by the sensory neuron, which stimulates an interneuron. This interneuron, shown here in white, integrates this information from the sensory neuron with other arriving stimuli and initiates step 4, the activation of a motor neuron. This motor neuron, shown here in black with its cell body located in the anterior horn of the spinal cord, sends action potentials to the periphery via the anterior root of the spinal nerve. Simultaneously, The interneuron may relay information such as pain to higher brain centers via axon collaterals. In step 5, motor neuron action potentials trigger the release of neurotransmitters, leading to the response of a peripheral effector. Here we see the signal branching to act on several skeletal muscle fibers. These muscle fibers, located in your arm, contract to withdraw your hand from the painful stimulus. In summary, a reflex arc consists of the stimulation of a receptor, activation of a sensory neuron, information processing in the central nervous system, activation of a motor neuron, and the response of a peripheral effector. So what? Why is it important to understand reflexes? Well, reflexes are incredibly important because they don't require you to pause and think. If you accidentally touch something sharp or hot, it's important to immediately withdraw to minimize tissue damage. While the pain receptors in your brain will be notified, which is how you interpret and feel pain, the reflexes occur independent of thought. Have you ever noticed how you withdraw your hand an instant before you actually feel pain? In addition, specific spinal segments control specific reflexes. So, testing the reflexes of a patient can tell a clinician the location and severity of spinal cord damage.
Brain and spinal cord are surrounded by 3 layers of connective tissue. These 3 layers are called meninges. The meninges consist of dura mater, arachnoid, and pia mater. Dura mater is the outermost layer, which is attached to the skull, while the innermost layer is called pia mater, which is firmly attached to the nervous tissue of the brain. Between these 2 layers, there's a third layer, which is called arachnoid mater. Between arachnoid mater and pia mater, there's a space called subarachnoid space, and cerebrospinal fluid flows in this subarachnoid space. Same applies to the spinal cord. You can also find the 3 layers of meninges from inside to outside. These layers include pia mater, arachnoid mater, and dura mater. You also have subarachnoid space between arachnoid mater and pia mater, where cerebrospinal fluid flows. Let's talk about the spinal cord now. So spinal cord is part of the central nervous system. It is located in vertebral canal. Spinal cord starts at the level of foramen magnum of occipital bone, and it ends at the level between first and second lumbar vertebrae. The end of spinal cord has a shape of a cone, and it is called conus medullaris. From the 2 sides of spinal cord, you have 31 spinal nerve pairs coming out, which provide nerves to upper extremity, lower extremity, and trunk. So although spinal cord ends at the level of first and second lumbar vertebrae, however, spinal nerves continue to extend in the vertebral canal, and they form a bundle, and this bundle is called cauda equina.
The spinal cord is composed of gray matter and white matter. Gray matter contains nerve cell bodies, dendrites, and the proximal parts of axons. White matter contains spinal tracts, which consist of myelinated axons. Sensory nerve impulses travel from the body to the brain through afferent nerves in the dorsal root ganglion and dorsal root. Motor nerve impulses travel from the brain to the body through efferent nerve axons in the ventral root. Dorsal and ventral roots merge to form each spinal nerve. The American Spinal Injury Association classifies spinal cord injuries according to spinal cord level and completeness of injury. A complete or level A spinal cord injury results in no sensory or motor function in the S4 to 5 spinal cord segments. Each spinal nerve provides sensory innervation to areas of the skin called dermatomes. A C4 to 5 level A spinal cord injury results in partial or total loss of sensation in the C4 and C5 dermatomes and total sensory loss below. Motor loss from a C4 to 5 level A injury results in tetraplegia or quadriplegia, which is characterized by partial or total loss of arm movement and total motor loss below. Structures with no motor function may include the diaphragm, requiring ventilator respiratory assistance. Acute treatment for a C4 to 5 spinal cord injury includes stabilization of the spine, physical rehabilitation, or surgical decompression of the spinal cord. Steroids are used to reduce inflammation. Physical and occupational therapy may be used to increase physical mobility and functional independence.
There are 31 pairs of spinal nerves, named according to their related vertebral levels. There are 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 1 coccygeal. Each spinal nerve is connected to the spinal cord by a series of rootlets. The rootlets on the dorsal surface of the spinal cord converge to form the dorsal root, and those on the ventral surface form the ventral root. A dorsal root contains bundles of nerve fibers carrying sensory or afferent information from the skin to the spinal cord. The cell bodies of these fibers are located within a swelling on the dorsal root, called the dorsal root ganglion. Each ventral root contains bundles of motor or efferent fibers, carrying impulses away from the spinal cord to the skeletal muscles. The cell bodies associated with these fibers are located in the ventral horn of the spinal cord. The dorsal and ventral roots unite to form a short spinal nerve in the intervertebral foramen. Because sensory and motor fibers of the roots mix together in the spinal nerve, they are known as mixed nerves. After emerging from its intervertebral foramen, each spinal nerve divides into 2 branches, called the dorsal ramus and the ventral ramus. Dorsal rami innervate the muscles and skin of the back. In contrast, ventral rami innervate the lateral and ventral skin of the trunk, the skin of the limbs, and the muscles of the trunk and limbs.
Spinal cord is part of the central nervous system, and it does have a role in processing the information. However, the most important function of spinal cord is to transmit impulses and information from peripheral organs to the brains, to and from the brains. So as the nerve enter or exit the brain through spinal cord, they create pathways or bundles which are called tracts. There are 2 important types of tracts we have. We have ascending tracts, which carry sensory information, and we have descending tracts, which carry motor information. There are more than a dozen types of ascending and descending tracts, but we only want to focus on 2 tracts. So the first tract is spinothalamic tract, which is a ascending tract. It carries sensory information of pain and touch from peripheral organs to the brain. And the second tract is called corticospinal tract. This tract is also called pyramidal tract. This is a descending tract. It carries motor information from the brain to voluntary organs such as skeletal muscles.